And so then this is pretty much our last section for bio/biochem techniques. Bio/biochem lab techniques for the exam, but tagging and functional experiments. So tagging, very common reasons that they use biological tagging is to really visualize and see where, so like in this image to the right, to really follow certain cells or certain molecules or proteins that are moving throughout an organism to track it. And so biological tagging is when a molecule is attached chemically to aid in detection of a biomolecule, such as protein, antibody, or amino acid. And a really common tag is GFP, so that's green fluorescence protein. And that is usually inserted downstream the promoter of a gene so that it ends up being expressed after transcription occurs from the RNA polymerase that usually works on the proper gene. And then so for the GFP, we usually pick specific genes of interest to visualize and track. And so then you're able to follow what happens as it goes through that process. And after tagging, GFP is one tag, but they can use a lot of different ones that they might mention in the bio/biochem passages. So, but just for the main function of tagging, that's what biological tagging is. And then functional experiments. So this is really used to study how genes and intergenic regions of the genome contribute to different biological processes. And the main functional experiments that they do are gain of function, so GOF, and loss of function, LOF. Gain of function is when there is an overexpression of the gene to determine the effects. So sometimes they might talk about an experiment where they added more expression of this gene and it caused like maybe another head to form from an organism or something like that. And then we have loss of function, which is the opposite of that, which is then you're taking away the gene expression to determine the effects. So then if you take away that gene to form the head, then the organism might form headless. So that's usually what their functional experiments are. And so this is the last question of this lecture. Okay, so we have here 100% saying A. This is a little bit, different. So the answer is actually B. So the question asks, which of the following DNA sequences is most likely to be recognized by a restriction endonuclease? So we know we're thinking restriction endonuclease, breaking up those fragments, so restriction enzymes. So here, maybe I'll make it a little bit, so yes. Okay, so here I'm going to visualize kind of what the sequences would be for each of these choices. So with A, we have this 5 to 3 prime, sequence here, and we want to determine then if the 5 to 3 prime sequence would be the same as it. So that's what the definition of the palindrome would be, if the sequence matches exactly its complementary sequence. So if we're thinking of complementary base pairing, you know that these two would be a T, and that would be a G, 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 G. And so we can see here that the 5 prime to 3 prime, that complementary sequence is T, T, G, G, T, T, which is different than the 5 to 3 prime in the original direction, which was AACCAA. So that's why answer choice A is not quite correct. But let's try it for this one, for choice B. So then we have the 5 prime here, where we have A, C, A, C, G, T. And then that's the 3 prime. So we see here that 5 prime to 3 prime in this forward direction is ACATGT. And then we see here in the complementary strand, 5 prime to 3 prime is ACATGT, which is the same exact sequence. And that's why choice B is the correct answer, with it being a palindromic sequence. For choice C, we could also do these just to make sure everything is right like that. But if we even are, I guess, starting off from this first C, so in this complementary sequence, it's saying that even from the 5 prime to 3 prime, it would start with a G. And this is already different from the forward sequence where this is starting with an A. So A versus G, and we already are able to kind of determine that that's not going to be the right answer. And then we go on to this 5 prime to 3 prime. So this one, the first letter starts off similar, with both 5 primes starting with a G. But then in the second one, we see that this ends up being a T and this one's an A. So that's why for choice D, it's also incorrect. And that's why choice B is the right answer.